So this part of the tutorial, we're going to talk about using HMM-Genie. So to use HMM-Genie, you're going to want to provide your own HMMs. So to do that, you're going to want to have HMMs to begin with. I talked about how to download HMMs from PFAM and TigerFAMs. So once you do that, let's say you want to have a custom HMM. You're interested in a particular gene. You couldn't find a good HMM for it anywhere online or in any public repository. So how do you go about making an HMM for this gene and then using that to profile a data set? So let's start. We're going to start with a gene called Psych1. So Psych1 is a diheme cytochrome. So there's the FASTA, amino acid FASTA file. So this is what the gene looks like. So Psych1 is implicated in iron reduction in the acetylthiobacillus ferrooxidans. It's considered part of the iron oxidation pathway. It's part of an Operon with another gene, Psych2. It's actually, this gene is already involved in P-Genie, so an HMM exists. But I'm gonna go through how I went about making that HMM. So we're starting with a FASTA file. So the first thing we're going to do is we're going to run phammer. So let's print the phammer command. Okay. So what phammer allows us to do is it allows us to query a protein sequence against a protein sequence database, right? So we're going to start with, we're going to provide it with a seq file, and that's going to be our Psych1. And we're going to provide it a database. And that database is going to be refseq. So to get refseq onto Jupyter, I subsampled it greatly. I think I subsampled at 0.1% to get it to a small enough file size where I can easily transfer it over to Jupyter. So it's not ideal, but it'll work. Well, it might not work. It might not get us an HMM that's good enough to be actually used, but for the purposes of going through the steps, I think it's sufficient. Okay, so one thing that we want to do is we want to create some output. What's the output going to look like? So I like this -- table output because it's gonna create a table out, and it's gonna show us what the hits look like, the annotations for those hits, and the bit score cutoffs and the e-values. So we're gonna call this psych1.refseq.tableout. And another important output is -a. And what -a will do, type this out. So -a is going to save multiple alignment hits to file. So all the hits to Psych1 that are present in this subsampled refseq database are going to be put into an alignment with Psych1. And we're going to be able to use that alignment to make an HMM, right? So I'm going to set an e-value cutoff of -e. -20, just to conservative. Actually, for all the Hummer commands, you're going to want all the options and the flags before the positional arguments. In the positional arguments, there's only two here, and that's the query sequence and then the reference database. Okay, so we have our table out. This is what we're going to inspect visually. We have our multiple sequence alignment that we're going to use to build the HMM. We have the e-value cutoff. We have our query, and we have our reference database. Okay, so we're going to hit enter, and it's going to search, and it's done really quickly because we only have 60 megabytes of refseq. Okay, so it created two files. It created the refseq table out. and we have a dozen or so hits, and we have an alignment in .com format. So when you get this output, you want to make sure to inspect it very carefully and call out the potential false positives, because you don't want to include genes that are potentially not involved in whatever process you're interested in the alignment. And you also ideally want to look at the alignment visually and make sure that it looks like a cohesive alignment, that there's nothing in the alignment that looks off, there's no errors in the alignment, and there's various different ways you could do this. I like a program called AliView, A-L-I. I'm going to type this out into the chat box. That's one of my favorite programs to manually inspect alignments and see if there's any sequences that look poorly aligned, or if there's any gaps that shouldn't be there, or if there's anything that looks off. I guess you could also use Genius. I don't have much experience with Genius, but I think that that's another good program where you can manually inspect. Adelaide recommends CView. Okay, I haven't used CView, but
yeah, I just wanted to say that this is a very important point. There are sometimes genes that are put into RefSeq or other databases that have been labeled as being a particular gene. And if they get into your alignment file, they'll have a lot of gaps, and they'll lengthen the sequence that's been aligned, and that's going to mess up your findings with HMM. So I don't know a better way than a visual inspection, and that really slows down the process. So I'm starting to play with a few ideas on how to have a manual alignment checker so that you can speed up this part of the process, because I have hundreds of genes I'm looking for innate immunity, so it's just very slow. I have to look at every single alignment file. I don't know, Arkady, you got any comments on that?
yeah, no, I completely agree. I'm well aware of this. The thing is called the transitive annotation catastrophe. Is if one sequence is annotated as something that it's not, and then it gets into an alignment, and you don't manually look through it and remove it, and then you include it in the HMM, that might make the HMM sloppier, and then it's going to attract more false positives. So I agree. I really don't know of a faster way to do it if you have a lot of sequences. When I was building the Fijini database, that's about 200 HMMs, and that took me 3 or 4 months to manually curate that database to make sure that all the sequences all the alignments were correct. So fortunately I can't give you an easy answer, and as far as I know, manual curation is. Oh yeah, so DanSpeth. I've read about DanSpeth has a really good bit score ratio filtering, and I think that was that published in your MCRA paper? I think I remember reading about that. Yeah, feel free to.
I've used it and published it in the MCRA read filtering paper, but it's an approach that originally came from comparative proteomics. The reference for it is RASCO 2005, I believe.
Yeah.
It uses the principle of comparing a sequence to, say, your target, and then to the larger database. And what you then have is the score against your target versus the score against RefSeq. And basically the question becomes, is your sequence as similar or as similar to your sequence of interest as it is to any other sequence in the database? And that's a way of getting rid of false positives or things that are perhaps like multi-domain proteins that do different things.
Yeah.
Dan, what was the author? Roscoe, R-O-S-C-O-E?
Yeah, Dan, can you provide the maybe not the link, or if you want the link, but the reference in the chat box so people could look it up?
Yeah, we'll do in a second.
Yeah, cool. Take your time. Okay, well, for now, let's move on. Let's assume for our purposes now that the alignment looks good, and we're ready to build an HMM out of it. So we're gonna run HMM build, and we're gonna provide. Well, let's see what the help menu looks like. Okay, so HMM build. So there's two positional arguments. The first positional argument is the name of the file that you want created. So you want this to be a file.hm. So this will be your HMM file. So we're going to call this psych1.hm. And then the second positional argument is a psych1 multiple sequence alignment. Okay, looks like that. So we're also going to provide a name for this HMM, and that's going to go in the one of the headers for the actual psych1 file, and we're going to call this psych1. Keep it simple. Run it, and there it goes. It's done. So we've created this file. It looks like this. This is basically the probability matrix. You got the name here. Yeah. Description. So this actually stems from the original FASTA file that we used to build this HMM. And so now we have this HMM, and we want to try our best to calibrate the bit score cutoff, or see what the bit score cutoff should be. So to do this, we're going to query our psych1 HMM back against the RefSeq database, or the subsampled RefSeq database, and just see what the results look like. This will tell us how sensitive the HMM is and how prominent it is in the database. And what I mean by that is, if there's certain proteins that are very ubiquitous among bacteria, you're going to get a lot of hits. And if there's any proteins that are not really commonly found and not well represented in the database, you're going to get a few hits. But let's just see what it looks like. So we're going to run HMM search -- table out. This is going to be what we're going to take a look at. And we're going to call this psych1.hm.refseq.tableout, sort of signify that we've used this HMM to query against a refseq, and this is what the table out looks like. And this will differentiate our other output, which is just refseq.tableout, which is our FASTA file queried against the refseq database. Okay. And I'm not going to provide a bit score cutoff or an e-value threshold just because I want to see what all the results look like. Okay. That was fast. All right, so you get a big table out like this, even with the small data set. We have a bunch of cytochromes in there that appear to have hits to our HMM. So yeah. So we see that top hits are all cytochromes. You get bit scores in the 270, 241 range. Then if you scroll down, we start getting things that look a little off. A lot of the times you get things that just say hypotheticals. Those aren't really, I wouldn't say those are very safe to use if you see a lot of hypotheticals. In this case, we see alcohol dehydrogenase, something that's annotated as an alcohol dehydrogenase. I'm not sure what that is. I think that in this case, there's really, I don't know, in this case, I'd be really hard pressed to find a optimum bit score unless I actually looked into each of these genes separately and did an alignment. For the purposes of this sort of tutorial, what I'm going to do is I'm going to say that's a false positive, and that's a false positive, and that's a false positive. And we're going to say, okay, everything above this looks like a cytochrome. Let's just provisionally make 60, 62. Anything above this, let's set that as the bit score cutoff. But when you see a continuous drop like this, that's really something that is more difficult to deal with. What's easier to deal with is if you have a bunch of hits with really high bit score cutoff, with really high bit scores, and then you see a steep drop off and you see false positives, and that's more of a clear cut. And another HMM that we're going to work with, MTR, I'm going to show you how that's the case. So that was a little bit easier for me to deal with. So I have a folder here called HMMs. In this HMMs, I have bit scores. And in here, I already typed everything out, so I'm gonna have to do it here. But yeah, so for Psych1, we're gonna go ahead and we're gonna set 60 for now. Let's go and look at another folder called MTRA. So MTRA is another cytochrome. It's a bigger cytochrome than Psych1. It's a decaheme cytochrome. It's encoded by Schwanella, and it's thought to be involved in ion reduction, right? So let's say for MTRA, you want to go a different route. Instead of having one sequence and then using phammer to gather homologs from RefSeq database, let's say you want to go about it a little bit more carefully. So what I mean by that is that you have a set of organisms that you know carry out ion reduction, and they're closely related to Schwanella. You have Pheromonas, you have Vibrio, Aeromonas. All of these are within the same family or order, I believe. And they all encode MTRA and they all reduce iron. So it's fairly, it can be assumed that MTRA is involved in iron reduction in this cloud of organisms, that given the fact that it's highly related. So we have this set of sequences that we've manually collected by hand, and we put them all in one file, and we want to use that to make an HMM without really going into RefSeq or any of those bigger databases to gather other sequences. So in this case, you're sort of limited by the diversity. You're limiting the diversity of the MTRA, but you are more protected against potential false positives, given the problem that Adelaide mentioned, is that sometimes people will submit stuff to RefSeq or NR that are misannotated. So you're avoiding that. But let's say you start with this multiple sequence file. It's not an alignment yet, so you want to build an alignment. So to build an alignment, it's really easy. We're going to run muscle. Muscle is really easy to use. I'll take 2 commands, MTRA, and then we're going to output mtra.fa. I like to use .fa as the alignment suffix. So we're going to enter. There it goes. Let's take a look at the alignment file. You're going to want to inspect this in AliView or CView or Genius, but let's just take a look and see what it looks like here in terminal. Okay, it looks great. We're not seeing a lot of really big gaps. All of these look like they're belong together. I'm not seeing any gaps actually. Oh, there are some gaps here. Yeah, not a lot of gaps. Pretty good alignment. And we're going to use this alignment to build an HMM. And we're going to do that using a similar command, same command actually. We're going to name this HMM MTRA. There it goes. Okay, so let's test it out. Let's run this HMM against RefSeq and see what the output looks like. All right, so there we have the table output. Actually, it's not an R, it should be RefSeq. We have our HMM, we have our database. So let's run that. There it goes, it's done. And we could take a look at the output. Not like that. Okay, so we have one really good hit, but it's annotated as cystathione reductase. Then we have cytochrome C. Okay, this is actually something, this is Schwanella putrefactions encode something called cystathione beta synthase, which is really closely related to MTRA. I'm actually very suspicious about this annotation. And what I would do is I would actually look at it further. And the only reason I say that is because MTRA is actually not annotated as MTRA in all the shoe and all the genomes. Sometimes it's annotated as something else. MTRA is also part of the nitrite reductase superfamily of proteins. Well, I don't know if it's superfamily, but it's a family of proteins, decaheme. So with something like this, I would definitely take a closer look at this and make sure that this is not a false positive or if this is a misannotation. But with something like this, with nitrite reductase, where I know that this is in the same family, you see that the bit score is really low, and I would shoot for way above that, probably closer to 481 after confirming what this actually is. I'm not actually sure what cystothione beta synthase is, but yeah. So in this case, what we're going to do is we're going to shoot for above 60s. So if this was the entire RefSeq data set, what we'd see a bunch of hits for Schumanella decaheme cytochromes, for the MTRAs, and that would give us hits in the ranges of 400, upper 400s, 500s, upper 300s, and then we're going to start getting into false positives and nitrite reductases, the DMSO family. That's not related to iron once we get closer to the 190 bit score range. So what we're going to do is we're going to set the bit score cutoff to be above that. Okay. So now that we have the HMMs for Psych1 and MTRA, we're going to put these in the same folder as the rest of the HMMs that I have preloaded on here. So we're going to move the MTRA HMM into the HMM folder, and we're going to move the Psych1 HMM into the HMM folder. So the HMM folder already had 3 HMMs, actually 4 HMMs that have been preloaded. So we have Psych2. Like I mentioned, Psych2 is normally found in the gene cluster with Psych1, at least in the known iron oxidizing bacteria. And MTRB and C are found in the same operon as MTRA, right? And we have catalase, just for fun. Catalase actually was taken from PFAM. So something that Adelaide asked me a while back is how you can tell where an HMM is from in Magic Lamp. So if you actually look in the HMM, you'll see that if you see a PF accession number, that'll tell you that it's from PFAM. If you see a TIG accession number, and the session number that starts with TIG, that means it came from TIGRFAMs. If you don't see one of those two, then it's an HMM that I made manually. But that supplemental table that I will publish with Magic Lamp will include all that information. Right, so if we look at the catalase HMM, we'll see that there's the gathering threshold, the trusted cutoff, and the noise cutoffs are all here. The description, the PFAM session. So if you want to look this HMM up in PFAM, you can copy and paste this into the PFAM search box, and then you'll find it. And you'll find all the associated information with it. Okay, so this is basically all the input that we need for HMM Genie. We have our HMMs. I made the bit score cutoffs file before this tutorial, but you can see that you basically set the first column as your HMMs and the second column as the bit score cutoffs. And I think we're ready to run HMM Genie and see what the command looks like and what the output looks like. Okay, so Magic Lamp, HMM Genie. Let me just refresh terminal. All right, I think we're having some technical difficulties because that's not supposed to happen. There you go. I think I included a colon that I wasn't supposed to include somewhere. Anyway, so there's the HMM pub menu. It looks almost exactly like the other ones, except that we have 2 additional arguments here, and that's the HMM directory and the HMM extension. So let's start building this command and see what it looks like. So we're going to provide the HMM directory first. That's going to be the HMMs. And we're going to make sure to avoid that bitscores.txt file. And we're going to say that the extension is HMM. So it just looks at the HMM files. We're going to have our bin directory. As data. That's going to have 4 genomes. And we're going to add the bin name extension. In this case, it's text. Should be FNA. That's not amateur hour over here. Okay. And then we're going to name an output file. Or name the directory that the output's going to be written to. Okay. So I guess we have the option of providing an e-value cutoff if you don't have a bit score cutoffs file. And we're going to add the bit score cutoffs file just because we have it. And we're going to run this and see what that looks like. Okay, so it's going to run Prodigal again because this is a new virtual terminal session, so the ORFs aren't written yet. So it's going to run Prodigal, and it's going to identify all the open reading frames. Like I said, if you have previously annotated open reading frames or gene calls, you can provide those and then add the -- ORFs flag. Another option that you have here is the -CLU, CLU. So I'll talk a little bit about this later on. Minimum size of gene cluster operon. So if you have an operon that you're interested in, if, for example, you have a bunch of HMMs for bioinformation, if you have the WISP operon, and you have 8 HMMs, and the operons normally comprised of 8 genes, and you want to just see the operons that have at least half of the operon complete. So then you provide -CLU4. And then it's going to filter out anything that just had a combination of 3 or 2 or 1 gene. So basically singletons. Or you can be as strict or as lenient as you want. I didn't provide a -CLU this time, so in default is 1. So it's going to tell us singletons. But let's see what that looks like. All right, results are written. Okay, this is the summary file. As you can see, we have a bunch of, we have the Schuanella MTR CAB operon. We have some singleton MTRC. We have a bunch of singleton Psych1 genes. And we have the Psych1 that is involved in iron oxidation since it's adjacent to Psych2. So this gene pair has actually been biochemically characterized to be involved in iron oxidation in acidithiobacillus. Right, so we know that this is a good hit, and these are potential false positives because they could just be cytochromes that happen to be related to Psych1 that aren't involved in iron oxidation. So one of the ways that we can filter them out is we could rerun the command, and then we can add -CLU 2. There it goes. So it's going to run. The output. And as you can see, we got rid of the other cytochromes, periplasmic cytochromes, and we just have Psych1 that's involved in the gene cluster with Psych2. Okay, hopefully that's clear. Okay, that's actually it for this tutorial. Unless there's any questions, or unless I'm forgetting something. There's the eval, there's the BAMs, everything's similar. Okay, yeah, so I wouldn't advise this if you have a collection of HMMs where not all things are in one operon. Like in our case, we have an HMM with catalase here. And catalase, there's no other HMM here that would form a gene cluster with catalase. So this got filtered out in the last run. So you'd want that as a separate sort of HMM collection, if you will. Yeah, okay. Okay, so if there's no more questions, I'm gonna end it here. Yeah, thanks.